So I forgot to add the disclosure slide, but I have nothing to disclose. Okay. So a significant component of lower urinary tract symptoms is due to failure of the nervous system to control bladder function, or it's because of failure of the neural pathways to compensate for bladder dysfunction. How the brain controls bladder filling and voiding remains, however, poorly understood. Okay. This is an image from the mouse brain atlas of a brain section in coronal view. The red circle is where the brain micturition center is located. In the pons, it's underneath the fourth ventricle and medial to the locus coeruleus. In the Pontine Micturition Center, here neurons are labeled with a tracer. Axons in red from these neurons project to the lumbosacral level of the spinal cord. Neurons in these spinal cord sites innervate to the trusor and the sphincter muscles. Today I'll focus on the role of CRH in micturition behavior. CRH expressing neurons are a subpopulation of glutamatergic neurons in the region. The CRH group of cells here is depicted as dark green dots. Corticotropin releasing hormone is a peptide stress hormone. It's synthesized and secreted by the paraventricular nucleus of the hypothalamus in response to a stressor. Besides in the hypothalamus, there are other regions in the brain where CRH is expressed. Here in the Micturition Center, for example, CRH labels a very select group of neurons. In our toolbox, we have video tomography. Using a thermal camera, we can record and assess mouse behavior. The camera hangs over these behavior cages and records the mice. So mice are continent, they go to the corner of the cage to void. On the thermal camera recording, the void is identified as a hotspot, which quickly cools off to ambient temperature. And after this, the void size, the location, and multiple voids in the same location, and other behavior can be analyzed. Okay. Using optogenetic light stimulation as a tool to manipulate neuron activity, we activated neurons to study their functional role. We inject very small volumes of adeno-associated virus into the mouse brain, and then we target and activate select group of cells in the Micturition Center now. In this schematic, you see a CRH targeting mouse line. Specifically, CRH neurons will be transduced with channelrhodopsin. And light stimulation is delivered to the brain through the implanted optical fiber. The cystometry trace here is only showing bladder pressure during one optogenetic stimulation event. The blue line represents the stimulation. So activating neurons causes bladder pressure to increase. And the left side of this graph shows light-evoked voids in 10% of the stimulations compared to control when mice are awake. And on the right side of the graph, you see in anesthetized mice that undergo cystometry, and that light stimulation evoked voids in 25% of the stimulations. Thus, optogenetic stimulation of CRH-expressing neurons in the PMC increases bladder pressure. We then wondered, are the CRH neurons necessary for bladder control and micturition behavior? In order to achieve complete ablation of the CRH-expressing neurons, we injected diphtheria toxin, DTA, on both sides of the brain. But first I'm showing a unilateral injection so you can appreciate the difference between injected and uninjected sites. The control site has CRH neurons labeled in green, and where the injection landed, which can be seen as the expression of the red label, there are no green cells left. So DTA ablated those neurons. Following injection with DTA, the fill-void cycle on cystometry was slowed. So usually a cycle takes 4 to 8 minutes. But when the mice did void, they were continent. So remember that CRH is a part of all the neurons in the PMC, so DTA ablation here is like a partial PMC lesion. We then sought to determine whether CRH neuropeptide released by the neurons may contribute to bladder control, as has been suggested by prior pharmacological experiments. Using the Crhis system, we knocked out the CRH peptide, but only in the PMC region. Selective disruption of the signaling, but leaving the neurons themselves and other neurotransmission intact in these neurons, has no effect on voiding frequency or voided volume in awake mice. Under anesthesia, when recording the bladder pressure, the frequency of voiding was unchanged. But also in this one example, the number of contractions not leading to a void are increased, and the compliance changed. So this could suggest a modulatory role of CRH on voiding contractions and voiding. So CRH-expressing neurons are a subpopulation of the glutamatergic PMC neurons. Optogenetic stimulation triggered a modest response, And deletion studies suggest that CRH expressing neurons or CRH signaling is not necessary for micturition behavior. CRH expression in a PMC may function to suppress the detrusor activity during the filling phase. So thank you, and I'd like to thank the members of the Zydel and the lower labs and other collaborators.
So an amazing amount of work to describe in 5 minutes, so fantastic. It is open to the floor. I'm very interested to try and work out from your data if what you're seeing is purely effect on bladder, or in other words, these are bladder-specific pontine neurons, or are you actually seeing a coordination with the sphincter? And I was thinking that perhaps the later cystometry with the lower amplitude non-micturition contractions could perhaps be indicating that you're not really seeing much effect on the sphincter.
Yes, correct. We think that the effects, what the CRH neurons do, their function, is related more to the detrusor activity than to the sphincter activity. That said, the CRH population is one group of all the glutamatergic neurons. There is another population. There was a recent publication by a group in San Diego. They are talking about the estrogen receptor expressing neurons, and they seem to be more in control of the sphincter.
That's fair enough. So that then makes it slightly more surprising when you're seeing that pattern of activity on cystometry, that you're reporting what you're describing as a continent pattern, that it's, I'm presuming what you're saying is big voids in well-defined locations. With that sort of pattern of CMG, are you not likely to be seeing a more distributed small void pattern, or am I misinterpreting, over-interpreting?
So the let's see, the fill void cycle is delayed. And we think that is because the glutamatergic neurons are also ablated. But you really do need more neurons to have the sphincter. I mean, the sphincter is still under control, let's say.
Okay. Yeah. Please, yes, of course.
Yeah, I have one question. Yoshimura, the University of Pittsburgh, so very nice talk. So this is optogenetic stimulation of this CRH, positive neurons, which can probably release that CRH somewhere in the spinal cord, right? So have you ever looked at the antagonist through the CRF receptors, which 1 and 2 is located sacral spinal cord? So in the combination, the intrathecal application of the CRH receptor blockade to look at the rust.
Yeah, we've never done any pharmacological studies.
Okay.
But Rita Valentino has described applying CRH and antagonists.
Two is more involved. That's what she said.
So I do expect that if you stimulate it optogenetically or
then applied to the CRF2 receptor antagonist, probably rebirth that condition of results. Probably that's what I'm thinking. So probably much interesting too.
Thank you very much indeed. Thank you.
Okay.